Hey guys, welcome to our lecture today on pathogenicity. So now that we all have a basic understanding of how microbes, can cause disease, we're going to look today at some of the specific properties of these microbes that contribute to their pathogenicity, and which is the ability to cause disease by overcoming the defenses of the host, and virulence, the degree and extent of pathogenicity. Please note that throughout the lecture, I'll be using the term host quite a lot, and that will most often refer to humans. If there are exceptions, I'll try my best to note those. And also note that, just a reminder, we discussed this a bit in the previous lecture, that microbes are not trying to cause disease. It's not a willful thing that they are deciding we will now make our host sick. The microbial cells are just getting food and defending themselves. They have no, greater awareness than that. So sometimes their presence, though, and their parts that they have and produce can induce symptoms in the host, and that is where the sickness comes from. So let's start off here with some terminology, starting off with the basics of what is infection. So this is the invasion and multiplication of microorganisms such as bacteria, viruses, and parasites that are not normally present within the body. Then we'll also be looking at pathogenicity, which is the ability to cause disease. Virulence is sort of a measure of pathogenicity because it's the degree of pathogenicity. And then just some other basic ones. A disease is a peculiar abnormal condition, a disorder of a structure or function that affects part or all of an organism. And then we also need to account for resistance. So what is resistance? So resistance is the inherent ability of an organism to resist harmful influences. These influences can include disease, toxic agents, or infection. And just a bit of a note here at the end that remember that these host microbe interactions are all forms of symbiosis, and that these symbiosis can typically be split into three groups when it comes to these pathogens or microbes in general. You have mutualism, which is beneficial to the host and beneficial to the microorganism. You have commensalism, which is sort of neutral or no effect on the host and is to the benefit of the microbe. And then you have parasitism, which is negative to the host and positive to the microorganism. And if you move down from mutualism to commensalism to parasitism, this is a shift towards disease processes, obviously, because you have the host, the host going from benefited to being harmed. Whereas if you move up the ladder, you have kind of a shift to establishment of a healthy host. Obviously, again, here you move from the host being harmed to the host being benefited. And so these shifting relationships, a healthy relationship will then essentially be a balance between a microorganism and a host, where disease is typically defined by an imbalance that is in favor of the microorganism. Here you can see that imbalance. So generally to cause a disease, a microorganism must be able to colonize a host. And obviously, if these organisms aren't able to, enter these host cells and do their thing, they won't be able to cause disease. They must also be able to multiply within the host. Obviously, they have to increase their numbers, otherwise there will just simply not be enough of them. Again, think of the small scale we're working with here. One single pathogen on its own will, pathogenic organism on its own is very, very unlikely to cause you any harm. And then lastly, it's unlikely to cause you any harm until it replicates, I should say. So they won't do anything to you, or it's unlikely that they'll do anything until they start multiplying and becoming a greater mass of cells. And then they also must, of course, cause damage to the host. Obviously, we are talking about pathogenicity. There are some exceptions, though. So if you look at something like Clostridium botulinum, which is the cause of botulism, if you eat food that has this microbe growing in it, you will get sick even if it doesn't grow within you. So it doesn't need this multiplication step, and not even necessarily the colonization. I mean, although when you ingest it, you could argue that they will colonize you. But even if they do not, you will still get sick because they have exotoxins. And the presence of these exotoxins make these microbes exceptions, because these toxins will induce a response in you, regardless of whether or not the organism is actually growing inside of the host. So once you are exposed to a potential pathogen, whether or not you will be developing disease depends on a number of factors. The first would be the dosage or the number of microorganisms. As I just said, the ingestion of small numbers of microbes, or the occurrence of small number of harmful microbes, very unlikely to cause an impact. You need quite a bit of these organisms, and then they obviously start growing and multiplying even more, and that's where you have your disease coming from. But your dosage can also impact your response time. If with greater dosages leading to quicker effects being noticed at times, although this isn't always the case, but it definitely is something you should be aware of. The number of microorganisms you ingest can definitely have an impact. You also have to account for resistance. Again, this is the ability of an organism to resist harmful influences. And these influences can include disease, toxic agents, or infection. And then you also, it also depends on the virulence, which is essentially the toxicity and the invasiveness of the organism, or can be broken down into those. So the virulence, just as a reminder here, is the degree of pathogenicity, where your toxicity is the capability of an organism to produce toxins. And the invasiveness is the ability of a pathogen to invade tissues. So to give you a breakdown of this, we're going to be looking at a couple examples. We'll be looking at tetanus, pneumonia, and the plague. So the ability to invade from tetanus is low, but its ability to produce toxins is high. You find the inverse in pneumonia, where its invasiveness is high, yet its ability to produce toxins is low. This results in both cases in a moderate virulence. But if you look at something like the plague, its invasiveness is very high, its toxicity is also high, which results in a very high virulence. I mean, we know that this thing absolutely, the plague smashed Europe and decimated the global human population, years and years ago. So essentially, you get to the point where true or primary pathogens have many virulence factors. And these virulence factors are essentially molecules that are produced by pathogens, which can include bacteria, viruses, fungi, and protozoa, that contribute to the pathogenicity of the organism and enable them to achieve the colonization of the niche of the host, immunoevasion, so invading the immune responses, and inhibition, immunosuppression, the inhibition of the immune responses, and entry or exit out of cells. So if in case the pathogen is actually intracellular, and it can also enable them to obtain nutrients from their host. So how do we measure virulence? So virulence can be measured, typically two measures. There's your ID50, which is your infective dose. This is the estimated number of organisms of a virus particles or of a pathogenic particles required to produce infection in 50% of normal adult humans or other hosts by a given route. So this gives you an idea of how of the virulence of the microbe. Then you also have LD50. I'm sure many of you have seen this before. So this is a lethal dose for 50% of the sample population. In other words, that is the lethal dose that 50% of the number of It is the number of organisms that is required to kill 50% of the hosts. So this is a measure of the potency. Sorry, I just misspoke. I lost my place a bit. So this is a measure of the potency. LD50 measures your potency of a toxin, where your ID50 measures the virulence of a pathogen. Okay, so moving on to portals of entry. So obviously we need these pathogens to enter the hosts, otherwise they will not cause disease. So there are three main portals of entry that we will be discussing. For the first is the mucous membranes. So many bacteria and viruses gain access by penetrating mucous membranes that line the respiratory tract and the gastrointestinal tract, or the urinary tract, or the conjunctiva, which is a delicate membrane that covers the eyeballs and lines the eyelids. Most pathogens, though, enter through the mucous membranes of the gastrointestinal and respiratory tracts, with the respiratory tract being the easiest and most frequently traveled portal of entry for infectious microorganisms. I mean, just think of COVID now as a nice example here. Microbes that are inhaled into the nose and mouth, drops of moisture and dust particles is essentially what you have to be concerned with when it comes to this respiratory infections. And again, I mentioned corona, but other examples here can include the common cold, pneumonia, tuberculosis, TB, the flu, and measles. The second portal of entry would be the skin. So first, you guys, I'm sure you're not aware at this point that the skin is the largest organ of the body in terms of both surface area and weight, and is our most important defense against disease. Unbroken skin is essentially impenetrable by most microorganisms, but some microbes gain access to the body through openings in the skin, such as hair follicles and sweat duct glands, where something like the larva of a hookworm can actually just bore through intact skin, and some fungi grow on the keratin in skin and infect the skin itself. Then we'll also be talking about the parenteral root. So the parenteral root is essentially other microorganisms can gain access to the body when they are deposited directly into tissues beneath the skin or mucous membranes when these barriers are penetrated or injured. So this is essentially anything like punctures, injections, bites, cuts, wounds, something that can result from surgery, or the splitting of the skin or mucous membrane due to swelling or drying can all establish these parenteral roots. Nice examples, or not nice, but examples here include HIV, hepatitis viruses, and then bacteria that can cause tetanus and gangrene can all be transmitted through this parenteral roots. So here we have some portals of entry of pathogens for some common diseases. We can see all the pathogens and the diseases they cause and how they enter the body. So here again, just as a reminder that the mucous membranes will include the mouth, nose, eyelids, the trachea, which is your windpipe, the lungs, the stomach, the intestines, the ureters, the urethra, and the urinary bladder. So all of those fall into this category of mucous membranes. But here you can see it is divided into respiratory tract, so that will be everything used to breathe, essentially, or gastrointestinal tract. So you definitely have a look at this. It's very good reference material for the exams and the tests that are coming up. So be sure that you have a look at these. Moving on here, we do arrive at the parenteral roots. So again, this is when microorganisms are deposited directly into the tissues beneath the skin or mucous membranes. So again, this needs some form of wound or puncture or swelling or something to occur for this to happen. A very nasty example here is the very first one, which is the disease of gas gangrene, which you shouldn't Google pictures of if if you're sensitive to stuff like that. It's really very bad. But that's also one of the primary things in wartime, where a lot of soldiers died to gangrene, and that was a result of open wounds that got infected that led to this gas gangrene eventually. Having soldiers have to have some of their legs amputated, because if you do not, the gas gangrene will spread and the person will die. Another one that is very, very well known is tetanus. This is the classic example of stepping on a rusty nail, and then you have to go for shots. So it's also a very, very well-known example of the parenteral root. Okay, so now moving on to the number of microbes invading the host. Remember that your dose, this dose, or here's an example of ID, so infectious dose, varying dependent on their portal of entry. So just some background here, we're going to be looking at an example of Bacillus anthracis. So this is the agent of anthrax, and is a rod-shaped gram-positive facultative anaerobic bacterium. Pretty cool that all of you know what that means by now, right? That is about 1 by 9 microns in size. It was shown to cause this disease. It was shown to cause anthrax. Interestingly enough, it was shown to cause anthrax by Robert Koch, which again is someone you guys should be familiar with at this point. So he took a blood sample from an infected cow, isolated the bacteria, and then put them into a mouse. So it's just some background there, and some more background on those organisms, something that is particularly, of import to you guys because you are Americans. So anthrax was actually used during the 2001 anthrax attacks, also known as Amerithrax, which is obviously a portmanteau of American anthrax from its FBI case name. So essentially what happened here, the attacks occurred in the United States over the course of several weeks, beginning on September 18th, 2001, which was 1 week after the 9/11 terrorist attacks. And letters containing these anthrax spores were mailed to several news media offices and to Democratic Senators Tom Dash and Patrick Leahy. I hope I said those names correctly. I apologize if I did not. And it led to the deaths of 5 people and the infection of 17 others. But something that you should be aware of here is look at how the number of endospores required to cause infection differs depending on how the anthrax actually, the portal of entry for the microbe, that the skin requires far, far fewer than inhalation, which in turn requires, again, far, far fewer than ingestion. So if you make contact on your skin with these spores, it's much more likely that you will get infected than if you inhale them or than if you ingest them. So something else that's interesting about anthrax, sorry, this is a bit of a tangent, but I thought I had an American example. I should probably include something from, a bit closer to home for me. So there was an interesting case regarding hippos. So during 2017 in Namibia, which is a neighboring country to South Africa, there was a drought that reduced the available their vegetation quite significantly. Thus, the only things that were really growing these small little plants that are close to the ground. And hippos, being vegetarians, would then wander out of the water at night and go forage, and they would actually eat these plants and then accidentally ingest some soil that could contain these anthrax spores. Anthrax is these spores can naturally occur in certain soils. And then what happened is you have hippos dying because they are infected with anthrax. And then because this was such a drought area and food was so scarce, these hippos would die, and then other hippos would eat those hippos while they are dying, when they've died in the water. So, they needed food, and they are omnivores, so they would eat their dead friends, essentially. And then they would also die because the anthrax would spread to them. So this led to mass die-off events where, many, many hippos died, and initially we didn't know what was going on. And it, after scientists investigated and figured it out, I just thought I'd tell you that story. It's a pretty interesting story. Okay, so something that you should also be aware of regarding the number of microbes is that, again, I'd like to stress that if only a few microbes enter the body, they will probably be overcome by the host defenses, so they won't cause real trouble for you. But if large numbers of microbes gain entry, the stage is very good for, the onset of disease. Specifically, if you look at something like quorum sensing, quorum sensing is a process of cell-to-cell communication that allows bacteria to share information about cell density and adjust gene expression accordingly. So this process will enable bacteria to express energetically expensive processes as a collective only when the impact of those processes on the environment or on a host will be maximized. Among the many traits controlled by quorum sensing is the expression of virulence factors by pathogenic bacteria. So essentially, when these microbes are just growing. Okay, first let's look at this. So first, I want you to look at these little symbols, which are pheromones, which are is essentially a pheromone comes from the Greek word meaning to bear an impetus. So it's essentially a secreted chemical factor that triggers a social response in members of the same species. So there you can see the pheromones, and there you can see way more pheromones. And these are pathogenic factors. So again, essentially what happens here is the microbes, when there are few of them, the concentration of pheromones would be low. So these microbes would not be changing, would not be expressing the pathogenic factors. However, once they recognize that, oh, wow, we actually have increased our numbers now, we have a nice high cell density, they will start expressing their pathogenic factors, and they will start attacking the host, which would obviously have much greater impact now because there are just so much more, so many more of them. So essentially their numbers would impact the concentration of the pheromones. And when the pheromone concentration gets high enough, the pathogenic factors will start getting expressed. So something else that we're going to chat about here is adherence. So almost all pathogens have some form of attaching themselves to host tissues at their portal of entry. For most pathogens, this attachment is called adherence or adhesion, and it's a necessary step in pathogenicity. So the attachment between a pathogen and a host is accomplished by means of surface molecules on the pathogen called adhesins or ligands that bind specifically to complementary surface receptors on the cells and certain host tissues. Here you can see the adhesin and the ligands, and they will bind to these receptors. And that is how the microbe will essentially stay in place. And here you can see pictures of that happening here. You see E. coli, the green on a human bladder cells. And here you can see bacteria adhering to the human skin. You can see these little bacteria over there. So it's also worth noting that microbes have the ability to come together in mass and cling to surfaces and take in and share available nutrients. These communities, which constitute masses of microbes and their extracellular products, can attach to living and non-living surfaces called biofilms. Remember the term biofilms because we will be discussing it in more detail in future lectures, and it's really, really important regarding microbial growth, even not just from a pathogenistic point of view, but also from a environmental point of view. Okay, so something else that we need to be aware of is your virulence factors. Again, these are molecules that are produced by pathogens that contribute to their pathogenicity and enable them to achieve the colonization of the host, essentially. So I would like you guys to recall here when looking at this picture that some bacteria make a glycocalyx that forms capsules around their cells. And this property will actually increase the virulence of the species. This capsule resists host defenses by impairing phagocytosis, the process by which certain cells of the body engulf and destroy microbes. And the chemical nature of the capsule appears to prevent this phagocytic cell from adhering to bacterium. So essentially, this capsule just means that the phagocytic cell just can't eat absorb, if you want to absorb or eat, if you will, the pathogenic organism. So one nice example here of a bacterium that owes its virulence to the presence of a capsule is in Streptococcus pneumoniae, which is the a causative agent of pneumococcal pneumonia. So some strains of this organisms have capsules and others do not. And the ones that have strains are virulent, but the strains without capsules are avirulent because they are susceptible to phagocytosis. So essentially the host defenses can just kill them and they'll never cause any problems. And then on this case, I just wanted to show you that, these adhesins bind specifically to surface proteins in the host cell. You can see there's some specificity over there. Okay. Moving on to enzymes and metabolic products, also falling into the category of virulence factors. So the virulence of some bacteria are thought to be aided by the production of certain extracellular enzymes or exoenzymes and their related substances. These chemicals can digest materials between cells and form or digest blood clots, among other functions. And why it matters if they break down blood clots is blood clots actually inhibit infection by preventing its spread throughout the host organism. So some examples here, you have kinases, which aren't listed here, but they digest fibrin clots. You have these high aluronidase, which digest polysaccharides that hold cells together. You have coagulases, which coagulate fibrinogen. You have hemolysins, which are lipids and proteins that cause the lysis of red blood cells and destroy their cell membranes. You have collagenase, which essentially breaks down collagen. And you have leukocidin, which is a type of cytotoxin created by some types of bacteria. So it is essentially a type of pore-forming toxin. And it gets its names by killing That's where the side will come from in the name, by killing leukocytes, which are white blood cells. Yeah. Moving on, we will be discussing some toxins and ecotoxins here. So toxins, just to give you guys a definition again, are poisonous substances produced by microorganisms. They can produce fever, cardiovascular problems, diarrhea, and a shock. And toxogenicity is the ability of a microorganism to produce a toxin. And then the first group of toxins we'll be talking about are exotoxins. So exotoxins are proteins produced inside the pathogenic bacteria, most commonly gram-positive bacteria, as part of their growth and metabolism. These exotoxins are then secreted into the surrounding medium during the log phase, during that growth phase. And these are typically soluble in bodily fluids, and they destroy host cells and inhibit metabolic functions. And then some other two definitions that you should be aware of is antitoxins. So these are antibodies that are formed against specific exotoxins, and toxoids, which are essentially inactivated exotoxins that are used in vaccines. And then yeah, so this is what you need to be aware of, because we'll be discussing endotoxins after this. So this is exotoxins produced in the cells that are then secreted out of the cells. Okay, now moving on to AB toxins. So AB toxins are essentially, they were the first toxins to be studied intensively, and are so named because they consist of two parts designated A and B. Pretty on the nose there. And both of these toxins are polypeptides. Most of the exotoxins that we know of are AB toxins, with the A part being the active, hence its name, enzyme component, and the B part is the binding component. So how does this AB toxins work? Essentially, in step number one, the AB toxin is released from the bacteria, essentially. Then we move on to step number two, where the B component attaches to a host cell receptor. So there you see the receptor and the B component. There's the AB toxin, and there's the B component attaching to the cell. The plasma membrane of the host cell invaginates, meaning it folds inwards. It's not really very illustrated very clear here, but essentially just folds in at the point where the AB ecotoxin and plasma receptor make contact, and the ecotoxin then enters the cell by receptor-mediated endocytosis. You can see a very good example of that. The AB toxin, moving on to number 4 here, the AB exotoxin and receptor are then enclosed and pinched off portion of the membrane. There you can see it still has a little membrane coating surrounding it. And then you can see at step number 5, the AB components of the exotoxin separate. The A component alters the function of the host cell, often by inhibiting protein synthesis, and the B component is released from the host cell. You can see there it goes, the B being released. And the receptor is inserted in the plasma membrane for reuse. A good example of AB toxins is botulism, which is essentially, again, this acts as a neuromuscular junction. The AB toxin acts in the neuromuscular junction, the junction between the nerve cells and the muscle cells, and prevents the transmission of impulses from the nerve cell to the muscle. The toxin accomplishes this by binding to nerve cells and inhibiting the release of the neurotransmitter called acetylcholine. As a result, the botulinum toxin causes paralysis, which makes muscle tone is lacking. So it's flaccid paralysis. So now we're going to be moving on to endotoxins. So endotoxins differ from exotoxins in a couple of ways. Endotoxins are actually part of the outer portion of the cell wall of gram-negative bacteria. Here, I want you to think back a couple of lectures that remember that gram-negative bacteria have an outer membrane surrounding the peptidoglycan layer of the cell wall. And this outer membrane consists of lipoproteins, phospholipids, and lipopolysaccharides, the LPS. And the lipid portion of the LPS, that is called lipid A, that is actually the endotoxin. So essentially, endotoxins are actually lipopolysaccharides, where exotoxins, which we just looked at, are actually proteins. Big difference there. And then the endotoxins are essentially liberated when the bacteria die and the cell breaks apart. So here you can actually see the endotoxins are normally just part of this external structure. And then when that external structure loses its integrity, or it bursts, or it lyses, these endotoxins are released. Note, though, that these endotoxins can also be released during bacterial multiplication. So it's not only when they're lysed, it can also be when the organism is just multiplying. So interestingly here, we have that antibiotics that are used to treat diseases caused by gram-negative bacteria can lyse these bacterial cells, and this reaction actually releases the endotoxins into the host body, and could initially lead to immediate worsening of symptoms, but that, those symptoms will usually go away when the condition improves and the endotoxins are broken down. These endotoxins exert their effects by stimulating macrophages to release cytokines in very high concentrations. So here you can see the impact of this these cytokines being released, or how that works. So essentially, your macrophage will ingest a gram-negative bacteria. So note that the endotoxin, this thing has endotoxins on its on its outer structure. And as the bacterium is degraded, it starts releasing these endotoxins, and this induces the macrophage to produce cytokines, interleukin-1, and tumor necrosis factor alpha. So once these cytokines, that's the important thing I want to focus on here, they are released into the bloodstream by the macrophages, and then they travel up to the hypothalamus in the brain, which is essentially, it's also where your body temperature is regulated. And once they reach the hypothalamus, they essentially reset the body's thermostat to a higher temperature, and that's why you have a fever. So they produce a fever. So, once these cytokines start spiking, they start becoming toxic, and all endotoxins produce the same signs and symptoms regardless of the species of microorganism, although not to the same degree. Very important there. So these include chills, fever, weakness, generalized aches, and in some cases shock and even death. And endotoxins can also induce miscarriage. So here, just a quick reference comparison between endotoxins and exotoxins. Here we have exotoxins, again, remember, produced inside and then secreted out, with endotoxins essentially being on the outer layer. They aren't produced inside the bacteria. And then something that I also wanted to point out here is look at the differences between the toxicity. You have a high toxicity for exotoxins and a lower one for endotoxins, and the lethal doses, where you only need a small amount of these exotoxins to be lethal, yet for endotoxins, they have to be much, much larger. Again, here we have those representations of, or representative diseases of gas gangrene, tetanus, botulism, diphtheria, and scarlet fever, where on the other side we have typhoid fever, urinary tract infections, and meningococcal meningitis. So just, it's a nice little way to look at these and to understand the differences between them. And then just a little funny slide for us to end off here. So today will be a much shorter lecture than normal. And then we will be moving on to some review questions. So true or false, LD50 is a measure of the virulence of a microbe. I'm going to give you guys about 5 seconds here. And the answer here is, of course, false. Remember, LD50, lethal dose, measures the potency of a toxin, not its virulence. Virulence is measured by the ID50. True or false, pathogenicity refers to the ability of a microorganism to cause disease. Is that true or is that false? I'll give you guys about 5 seconds. The answer here is, it is true. That is exactly what pathogenicity refers to. True or false, exotoxins are proteins produced inside pathogenic bacteria. I'm going to give you guys about 5 seconds here. and this is true. Some things that I want to point out here is keep in mind that there are in fact proteins. That's very important. So that, you know, if I change that to something else, that would have made it false. Produced inside is the other big thing that differentiates exotoxins and endotoxins. So definitely I want you guys to pay attention to little details like that, because those are typically the types of things that I could change to make this statement false. So as a tip there for future tests and exams. So I know a little multiple choice one to finish off here. Which of the following is not a portal of entry for pathogens? The mucous membranes of the respiratory tract, the mucous membranes of the gastrointestinal tract, the skin, the blood, or the parenteral root? Which one is not a portal of entry for pathogens? Can you give you guys 5 seconds again? If you need some more time, feel free to pause the video. The answer here is, of course, blood. Hey guys, that'll do it for today. I hope you guys enjoyed the shorter lecture. We are also coming up on the second class test. Just a couple of notes on that. It'll be the exact same format of the first class test. Keep in mind that they are non-cumulative. My exams and all my class tests are non-cumulative. So the material covered will be the material following the midterm exam. So only those couple of lectures we've done since then. And it'll also include the next lecture, of course. So just keep in mind that those were, that will be your content. It'll be the exact same format. So open book, lockdown browser. So again, guys, just prepare for that, and then good luck. I'll see you guys in the next lecture.